morning to you all. Welcome once again to Pedagogical Content Knowledge in Biology. Now, this is session 2 of Unit 3, and in session 2, we are going to know a summary of biology skills, some skills in biology, okay, which has been summarized for this session. So, the objective for today's lesson is at the end of the session, you'll be able to explain the skills using your own words. Using your own words, the skills that I'll take you through, I expect that you should be able to explain in your own words. And then you discuss the biology skills mentioned in their various groups. Okay, so now equipment handling. Now, when we say equipment handling, I'm talking about proper equipment handling, okay, and use of tools and equipment for practical and experimental work. So the teacher, you should ensure that students acquire a high level of proficiency in the use of tools and equipment for scientific work. Some students don't know how to handle laboratory equipment. So you as a teacher, when you take students to lab, all the time I tell my students that if you take students to the lab, you expose students to the equipment in the lab. After you've done with the theory aspect of the equipment in the lab, you come and do the practical aspects by exposing them to the equipment. You tell them the functions of the equipment, how it is being handled, how you handle microscope. Some of these equipment in the biology lab are microscope, hand lens, petri dish, slides, models of standard objects or standard materials. You see models of standard materials, and then other materials. Though there are a lot of equipment in the biology lab, so you have to demonstrate how you handle it in the lab for your students to also have a feel of that. The next one is planning and design of experiment. Planning and design of experiment. When you are conducting an experiment, you have to plan. Okay, you have to put an experiment. You have to plan what you expect students to achieve at the end of the experiment, materials needed for the experiment, the title of the experiment, aims and objectives of the experiment, processes involved in the experiment, okay, conclusion, discussions, all those things you have to plan on paper. You have to plan on paper before you come out to inform students about how the experiment will go. Okay, the next one is observation. Observation, some students don't know how to observe specimens. When you say you should observe the posterior view, you'll be looking at the anterior view. So all the time, before you even teach observation, expose students to how to observe, you have to teach them body symmetry, the symmetry of an organism, okay, how the body is being positioned, okay, symmetry of an organism, okay. So you have to teach students that you have the radar symmetry, bilateral symmetry, and then you also teach them the posterior view, anterior view, all those things you have to teach the students, so that when you send them to the lab and you say observe the anterior view of an insect, of say a cockroach, so the students should be able to do that. The next one is manipulation. This one is a hands-on activity. Manipulate. How do you manipulate the microscope to observe the specimen on the slide? And this one, you teach students the theory aspect of the functions of the parts of the microscope. Okay, you teach the students, and then you teach them the functions of the parts. You bring them to the lab. You demonstrate how to observe specimens under the microscope. The next one is classification. Classification is the grouping of organisms according to their characteristics. So you first teach topic classification before you bring students to the lab to conduct experiment or practicals. Drawing, you teach students how to draw, how the labeling should be, no dotted dashes. You expose students to these skills. Measuring, the skill of measuring. Students should be exposed to skill of measuring, how to measure, so that they'll be able to transfer this knowledge in their exams. And then interpretations. How do you interpret? How are you going to interpret that after your practicals? How do you interpret, okay, from your findings? And then the next one is recording. How do you record figures? How to record figures? And then reporting. How to report a practical or a field work. How do you report it? How to report it? And then you write your title, your aims and objective, process, procedures. All these are steps in reporting. The next one is conduct in laboratory. Conduct in laboratory field. So we teach students how to conduct biology experiment or practicals. Teach them when you go to the field how they should behave and all that. So these are skills students should acquire in order for them to gain understanding and then construct their own knowledge, then transfer the knowledge. Okay, so we've learned a lot in this session. We've learned a lot in this session, and then brought the lesson to an end. So we are trying to review what we've learned. So in reviewing what we've learned, I want you students to answer these questions. I want you to answer these questions. So question, I'm saying you should discuss 5 practical skills required for effective practical and experimental work in biology teaching. Discuss 5 practical skills required for effective practical and experimental work in biology teaching. So you discuss it on any available e-learning platform, and then you come out with solutions.